Welcome to this video. In this video we're going to talk about cataracts and cataract surgery. So let me begin by giving an outline for the structure of this video. So we're going to start off with a revision of the anatomy of the eye, because it's really important that we understand that in order to understand cataract surgery. And then what we're going to do is we're going to specifically focus in on the microanatomy of the lens, so we'll have a look at the histology of the lens, and then we'll talk about the physiology of the lens briefly, i.e. what the lens usually does in the eye. Then what we'll talk about is what a cataract is, and we'll then move on to talking about cataract surgery. Right, so we'll begin then with, as I say, the anatomy of the eye, a revision of the anatomy of the eye. So the way I'm going to do this is I'm going to draw a picture showing all of the different layers of the eye, starting from the outer side and then going into the inside. So what I want you to imagine is we're going to draw a transverse section of the right-hand eye. So imagine taking a horizontal plane through the right-hand eye, and we'll level this horizontal plane at the correct level such that we will catch the optic nerve as it leaves the back of the right eye. So in our transverse plane then I'm going to be able to draw the optic nerve. Okay, so let's get on with drawing this then. So a good place to start is by drawing a circle. So here we go. So this is the outer wall of the right-hand eye, and as I say, we've taken a transverse plane, so a horizontal plane through the right-hand eye. So here we go. So this is the outer layer then of the wall of the eye, which is of course the sclera, and we'll have the optic nerve coming out the back on the nasal side here. So just to orient you, this is the anterior portion, so this is facing the outer world. This is the posterior portion facing the back of the orbit into where the brain would be. This is lateral and this is medial, so the nose would be here. So it's on this side that you'll have the optic nerve coming out the back like so. Okay, so here we go. Right, so at the front then, of course, the outermost layer of the eye is the cornea, and this will extend forward, so I'll draw this now. So here we go, here is the cornea. Right, now what I'll do is I'll actually turn that from being an infinitesimally thick line into having an actual thickness here to make this outer wall of the eye. So at the front, the outer wall of the eye is the cornea, and the cornea covers the iris. So in fact, I'll just draw a picture that you're probably very used to seeing of the eye, because this is what you usually see when you look into someone's eyes. Okay, so here is what you would usually see of someone's eye, and you'd usually see the iris, which is the coloured portion here, the coloured muscle, and then with the pupil in the centre, like so, and then you usually see the white of the eye here. The white of the eye is the outer layer of the eye for most of the eye. It's the outer surface of most of the eye, and it's known as the sclera. So I'll just put that key word here. However, there is effectively a continuation of the sclera over the iris here, and there that continuation is known as the cornea. So here is the cornea, and the cornea, unlike the sclera, is transparent. So the sclera, of course, is white. The cornea is transparent. So I'll just finish this up here. So here is the sclera coming round on the lateral side here, like so, and it'll go back. So the sclera is continuous with a covering over the optic nerve at the back here as well. So not only is it effectively continuous with the cornea at the front here, so this is the cornea that I'm colouring in yellow here, so that's the cornea, but it's also continuous with a covering over the optic nerve that will protect the optic nerve at the back here, like so. So I'll colour this all in as one yellow continuous layer here. Okay, so that's the cornea, the sclera, and the covering of the optic nerve at the back. And that's the outer layer of the eye. So all of this portion would appear white all the way around if you were to take it out, and you can just see portions of it here. So this portion here, and in fact I'll colour it in. So we'll have it coloured in yellow here. And now, of course, usually it would be white, it shouldn't be yellow, that would be indicative of jaundice, but never mind. For the basis of this picture, the sclera is being coloured in yellow, so this portion here is effectively equivalent to this portion, and this portion here is equivalent to this portion. So I'll remind you that we've taken a transverse plane, so we've effectively cut the eye like so, and we are now looking at the open section of the eye. Right, so next up, what shall I add on to my picture next? So I think I'll put on the iris next. So the iris is this muscle with the pupil in the middle that sits behind the cornea like so. Okay, so of course, in reality it's a circle of muscle, like so, with the pupil as a whole in the middle. But of course we're just looking at a transverse cross-section, so we're not going to see it as a circle, we're just going to see two little strips like what I've drawn here. So I'll just colour in the iris in blue here. Let's suppose our person has blue eyes, and this is the iris here, with the pupil in the centre. Okay, so I'll add on the word iris. And I'll also label up the pupil as well. So this hole in the iris is called the pupil. Now, you can see that there is a chamber in between the cornea anteriorly and the iris behind. This chamber has a special name. It's known as the anterior chamber of the eye. So that cavity in between the cornea anteriorly and bounded by the iris behind is known as the anterior chamber of the eye. And it's filled with a fluid known as the aqueous humour, and I'll talk in a moment about where the aqueous humour is actually produced. So we'll see the ciliary body is the production site for the aqueous humour, and we'll add that onto the picture in just a moment. So this space here, which I'll just colour in pink rather, is called the anterior chamber, and it's full with a fluid known as the aqueous humour. Right, so that's the anterior chamber and the aqueous humour. Now let's go further back. So next what we're going to see is the ciliary body, which has two functional components. It has a muscle component and it also has a secretory component. The secretory component is going to produce the aqueous humour. The muscle component is going to be extremely important in controlling the shape of the lens, and is actually going to be extremely important in accommodation, which is changing the shape of the lens so that we can change the power of the lens, and this is what allows us to focus light coming from sources that are differing distances away from the eye. And we'll discuss all of that when we come on to the physiology of the lens later on. It's very, very complicated, but we'll go through it later on, at least to some degree. So I'll draw the anatomy is very simple though, so we'll draw the ciliary body here. So here is the ciliary body, and it's effectively continuous with another layer of the wall of the eye, which is this layer here known as the choroid. So I'm drawing on the choroid now. So this lump at the front, and it's actually, again, it's circular, so it would be a circle structure all the way around. So I could have actually taken this plane like so, and it would have still looked the same. Indeed, the iris would have still looked the same. In fact, everything I've drawn so far, apart from the optic nerve, would have looked the same if I'd gone in that sagittal plane like so. So this is a circle, and in fact, how can I show this better? I'll draw this on here. So there's effectively behind the iris, the ciliary body forms a loop like so. So again, we are just seeing a cross-section of it, which is why it looks like so. Okay, and then it's continuous with this layer of the wall of the eye that is underneath the sclera here, and which doesn't go into the optic nerve. So you can see I'm terminating it before we go into the optic nerve, and this is called the choroid. So I'll just straighten this back up. Let me now colour in the different portions here. So here, this is the ciliary body, here in red, and I'll continue it on with the choroid, which I'll also have in red here, like so. So this is the choroid extending backwards, and likewise on the medial side here I'll do the same. So, to label this up, anteriorly this is the ciliary body, and I'll remind you in a moment about the two functions of the ciliary body. And this layer here is the choroid layer of the wall of the eye, and that goes all the way around the wall of the eye here, except it doesn't go into this portion where you've got the optic nerve coming off, as I've shown in the picture. Okay, so now let's go back to the ciliary body. So one of the functions of the ciliary body is secretory, to produce the aqueous humour that's in the anterior chamber. The other function is in controlling the shape of the lens, and the way it does this is the lens is going to be positioned here. So I think we now need to add on the lens. So here is the lens of the eye, like so, and it's suspended, attached to the ciliary bodies, or the ciliary muscles if you like, by ligaments known as suspensory ligaments. So these lines that I've drawn here, these are the suspensory ligaments. So just to put the key words down, so these are the suspensory ligaments. And again, the suspensory ligaments are going to form a circle effectively. You're going to have suspensory ligaments going all the way around. If I just put it on this picture here, all the way around from the ciliary body, and of course you can't see this, you'd have to be able to take away the iris and the sclera to be able to see this. This is underneath. You've then got the suspensory ligaments going all the way around with the lens then suspended in the middle. So those are suspensory ligaments, and because they form this loop, the whole loop of suspensory ligaments is often referred to as the zonule of Zinn. So these are two terms that you might hear for the same thing, the suspensory ligaments, or you can call it the zonule of Zinn, this structure that suspends the lens in the middle of the ciliary body effectively. Okay, so just adding on the keyword lens as well, which is of course the smarty shaped thing in the middle there. Okay, so the ciliary body is going to control the shape of the lens. The lens can either have a shape like this, where it's very stretched out and not particularly globular, or it can have a shape more like this, where it's not very stretched out but is incredibly thick now. And these different shapes, they are going to focus light in different ways. They're going to be different power lenses, and I'll come on to the concept of the power of a lens later on when we talk about the physiology of the lens and what the lens is doing, i.e. how it focuses light and all of that. It's a complicated concept, but we will do it. It is the ciliary body which controls whether the lens looks like this or looks like this. Now, the ciliary body, when it is relaxed, it's going to be quite a large circumference loop of muscle. So if you imagine, when this contracts, it constricts the whole, basically. When this muscle contracts, what's going to happen is the loop's going to move inwards, and the diameter of this loop of muscle is going to reduce. Therefore, the suspensory ligaments are going to go slack, and the ciliary body is going to become like this. So when this constricts, the lens, sorry, I said ciliary body, I meant lens. The lens is going to end up looking like this. Whereas when the ciliary body is completely relaxed, the diameter of this circle will be much larger. So imagine, that this is a ring of smooth muscle cells. You've got smooth muscle cell after smooth muscle cell after smooth muscle cell, and indeed that is what it is. If you imagine having such a ring of smooth muscle cells, and I will just draw a little picture of this over here. So if you've got, here's a smooth muscle cell, here's a smooth muscle cell. It's just like if you've ever thought about how the smooth muscle cells around the arteries or arterioles work to constrict arteries or arterioles, or even bronchi and bronchioles as well. So here we've got a ring of smooth muscle cells. If they were all to contract, then all their lengths would shorten, and if all their lengths shorten, then that means the whole circumference of the loop is going to reduce down, and when the circumference of a loop reduces down, the diameter of that loop is going to reduce down, so it's going to constrict the diameter of this loop of muscle. Okay, so that will then mean that the suspensory ligaments will go slack, and that will mean that the lens goes into that sort of a shape. Whereas when it's relaxed, it will be a much larger diameter loop, and the suspensory ligaments will go taut, and they'll pull the lens into this sort of a shape. Okay, so the ciliary body has this role in controlling the shape of the lens, and we'll talk about why controlling the shape of the lens is so important for accommodation later on when we come on to the physiology. Now, you can see then that there is this space, another chamber, which is bounded anteriorly by the iris this time, and posteriorly by the lens, the zonule of Zinn, and the ciliary body. Okay, and this chamber is known as the posterior chamber, and it is clearly continuous with the anterior chamber via the pupil, so they're connected to one another. And indeed, the fluid in the posterior chamber of the eye is exactly the same as the fluid in the anterior chamber. How could it not be? It's aqueous humour. And indeed, where does the aqueous humour come from? It's coming from the ciliary body. So the ciliary body secretes aqueous humour into the posterior chamber. It goes through the pupil into the anterior chamber, and then it actually gets reabsorbed at this sort of edge between the cornea here and the iris, so in this little corner, if you like, all the way around, because of course everything's a circle again, so all the way around that edge, that's where there are vessels that are going to reabsorb the aqueous humour, so there's a continuous cycle there. Okay, right, so that's the posterior chamber. Where to next? Well, next I'll talk about what this massive great chamber is here, and this is actually a confusion point for a lot of people. A lot of people, very intuitively, would say this is the posterior chamber of the eye, and that all of this is probably just the anterior chamber. But no, the posterior chamber is the one that I've talked to you about. This is the posterior chamber, this tiny little chamber here, and this is the anterior chamber. This massive great chamber here, the main chamber of the eye, if you like, this is the vitreous chamber. Okay, and it's not filled with aqueous humour, instead it's filled with vitreous humour. So it's filled with something known as the vitreous humour, which is jelly-like in consistency rather than constituency. Consistency is what I meant. Okay, so this is the vitreous chamber here, and it's filled with the vitreous humour, often just referred to as the vitreous. Right, further anatomy that I want to discuss then is obviously where is the retina. That's the main part, that's where the photoreceptors are, and we haven't yet put that on. So now, over the top of the choroid then, you're going to have the retina. So I'll draw this on here, and the retina is going to contain photoreceptors, and it also contains a huge number of nerve cells that are going to conduct the information that the photoreceptors have gathered to the brain. And this is why I'm about to draw it going into the optic nerve. Of course, the photoreceptors don't go into the optic nerve, but the nerve fibers do go into the optic nerve. So often in pictures, it's practice to draw the retina as being continuous with the optic nerve in this way, because indeed the neurological tissue of the retina, which is a huge amount of the tissue of the retina, is continuous with the neurological tissue of the optic nerve. Okay, so here is the retina then, and here is the nervous tissue in the optic nerve, like so, all coloured in orange. And I'll remind you again that the photoreceptors, those are part of the retina, the bits that the cells that actually detect light, and they generate electrical signals from detecting light. However, another key component of the retina is all of the nerve cells that are going to receive the information from the photoreceptors and transmit it to the brain, and these are going to send their axons into the optic nerve here. Okay, so let's just label this up. So this is then the retina here, and I'll underline that in orange. And then over here we have cranial nerve 2, which I haven't so far labelled up, the optic nerve. Okay, finally let me point out that I have left this gap down the middle of the optic nerve. That is there is a canal down the middle of the optic nerve known as the central canal of the optic nerve. So this is the central canal of the optic nerve, and in that central canal of the optic nerve is going to run the central artery of the just the central artery, or you can call it the central artery of the retina. So in red there I have drawn this artery coming in. So in the central canal you're going to have the central artery, and this is the artery that's going to supply blood to the retina. So it arrives at the back of the eye, where the optic nerve is, and indeed you can actually see the point where the optic nerve is on the back of the retina. If you look at the retina using an ophthalmoscope, you can actually see a disc on the back of the retina, which is where the optic nerve joins the eye, and that's known as the optic disc. Another little key expression there. And then radiating out from the optic disc, you can see arteries that are going to supply the retina, and they actually go over the top of the retina. And they're all coming from the central artery that's arrived with the optic nerve, or in the middle of the optic nerve. And indeed, as I say, they are on top of the retina, so really, you might ask, well, why don't we see them if they're on top of the retina? Because they're going to be blocking light getting to the photoreceptors that are underneath them. And the reality is that we do see them. Well, our brain subconsciously sees them, but conscious portions of our brain have managed to not pay attention to them, so we're not usually aware of them, but our eyes are detecting them all the time. We are seeing them, we're just not consciously seeing them. Okay, right, so there is the anatomy of the eye. Next, what I want to do is go over the microanatomy of the lens in more detail than we've discussed it so far. So at the moment, I've just put the lens on as a Smarty-shaped thing there. And you know, from basic biology, you will be aware that the job of the lens is to focus light onto the back of the retina. But let's now actually discuss the structure of the lens in more detail. What is this thing made up of? So, there are two main parts to the lens. There is an outer portion, which is known as the capsule, and then there is an inner portion, which is just known as the centre of the lens. So let me now draw the lens larger here. So here we will have our lens. Right, so the outer capsule is a tough capsule of connective tissue. So many different organs have capsules. So for instance, the kidneys have capsules, the adrenal glands have capsules. These are just connective tissue layers that keep everything together and protect everything. Okay, so this capsule of the lens is just going to be a thin but tough connective tissue layer that is going to contain the centre within it. So I'll colour in the capsule here in blue. So this is going to be the lens capsule. Whoops. Okay, so this is the capsule here. Okay, so then everything that's inside the capsule is going to be the centre of the lens. And actually, let me just add some more information onto the capsule. The capsule is made out of lots of connective tissue, and principally one of the major types of connective tissue that is forming the capsule is type 4 collagen fibres. So there are loads of different types of collagen. All it means is the exact proteins that are used to make the collagen. So there are loads of different proteins, loads of different polypeptides that you can use to build collagen fibres, and the different types of collagen fibres are built out of different combinations of these polypeptides. So all type 4 collagen means is a specific combination of polypeptides has been used to make those collagen fibres that have made that connective tissue. So principally the capsule of the lens is made up of type 4 collagen. Okay, then the portion inside the capsule is then known as the centre of the lens. Now what does the centre of the lens consist of? Well, the centre of the lens is going to consist of things called lens fibres. And you might think, well, are these just some sort of connective tissue fibre? But they're not actually. They are cells that have lost their nuclei, that have lost their other organelles, and which have become filled with a certain type of protein known as crystallins. And in fact, the concept here is very similar to the concept of the keratinocytes of the skin. So if you're familiar with the biology of the skin, the epidermis in particular of the skin, you'll know that there are stem cells at the base of the epidermis which continually divide and produce progeny, some of which will remain as stem cells and maintain the population of stem cells on the base of the epidermis, and others which will move upwards in the layers of the epidermis. And as they move upwards, they differentiate and they become gradually more and more close to the keratinocytes on the surface. And as they go through this differentiation process, they lose their nuclei and become utterly full with keratin, and then just become these sort of shadows of cells that are just full of very tough fibres of keratin, and this makes the outer layer of the skin very tough. So, the lens fibres are a similar concept. You're going to have some cells which are normal cells, i.e. they have intracellular organelles, and these are going to produce these lens fibres. So, let me firstly talk about the cells that are the normal cells with intracellular organelles that are going to produce the lens fibres. So, these are produced by the lens epithelium. So I'll show you the lens epithelium here. So on the anterior surface of the capsule, so this is the anterior capsule here, and this is the posterior capsule. So for obvious reasons, this is just facing the anterior side of the body, and this is facing the posterior side of the body. Now, on the anterior capsule, but not on the posterior capsule, you have what is known as the lens epithelium. So this is a epithelium consisting of cuboidal cells, which are going to produce the lens fiber cells. So this is the lens epithelium. Right, so these are normal cells. They will have intracellular organelles, and they're going to divide, and their progeny will become either lens epithelium cells, or they can become lens fiber cells. Now, unlike the skin, this division process and production of lens fiber cells is extremely slow. In the skin, the turnover is very high because you're continuously having to replace the cells that are being lost to the environment. In the lens, it's very, very slow because you're not losing lens fibers. In fact, the lens fibers that you produce, you will have for the rest of your life. In your lens at the moment, you will have lens fibers that were produced long, long ago when you were an embryo in your mother's womb. So you never lose your lens fibers, so you just accumulate more and more inside the lens. So for that reason, in adult life, the actual production of new lens fibers has to be extremely slow, otherwise you're going to end up with problems with lack of space for new lens fibers. Okay, so the lens epithelium, it is the bunch of cells which are responsible for producing lens fibers, and throughout your adult life they will produce some new lens fibers, but the turnover is very slow, nothing like what you've got present in the skin. Okay, so now let me actually talk to you about what a lens fiber looks like. So they're very long cells, and they have one of their ends long and narrow, and one of their ends faces the anterior capsule, and the other end is going to face the posterior capsule, and they're not just in a straight line, so it's not like this. Okay, so this is what my lens fiber is going to look like, but it's not positioned like that. You can see I've bent it round like this, and this is the way that lens fibers are going to be positioned inside the lens. They're always going to be sort of circumferentially. They're always going to be sort of like forming an arc of the circle around the centre, and this gives the lens a sort of onion skin structure, because on this side the lens fibers will be oriented like so, and then we'll have another one maybe like this, another one like this, another one maybe like this, another one like this. I hope you get the picture of this. So these cells have no nuclei, which is why I haven't drawn nuclei on them, and I'll just colour them all in orange, green here, to show that they're lens fibers. And instead they are absolutely full up with special proteins known as crystallins. So unlike the keratinocytes of the skin, they do not fill with keratin. Instead, they fill with special proteins known as crystallins, and these proteins help to keep the lens fibers transparent, because of course the lens does need to be transparent. It needs to allow light to move through it, and they also help to bend the light, and of course that's the function of the lens fundamentally, to bend light. So the crystallins are believed at least to help with the bending of light. Okay, so I think that's all I want to say then about the histology of the lens. I think we'll have a break here, and in the next video what we'll move on to is discussing the physiology of the lens, and we'll do the basics of this, but enough that you have at least some understanding of it. So we will discuss things like what is meant by the power of a lens, to give you a basic understanding of what the lens is doing. But optics is incredibly confusing, so we're not going to go into it in the fullness that physicists would go into it.